So I hope everyone had a nice lunch, reflection on this morning's discussions. So I've got 10 minutes to give you a whistle-stop tour of how we're going forward to use the increasing knowledge of the biology that underlies childhood cancer to improve how we decide who gets which treatment now, and also how we use this information to develop some new therapies. But the following speaker, Peggy Brock's going to talk a bit more about that. So really just where are we now? This is data published just before Christmas from the Oxford Office for National Statistics. You can see now that 5-year survival, which generally equates pretty much to cure for many childhood cancers now, so just over 80%. This is English data. And the remaining challenges are really the high-risk tumours that we don't know, we don't have effective enough treatments for. I've just listed a few there. And then the good risk ones where we've got effective treatment, but we're probably over-treating some children, and sometimes we're not we're not recognising the bad risk children early enough, and maybe they wouldn't be so bad risk if they got slightly more intensive treatment at the beginning. So this is the sort of traditional approach to diagnosing a child with cancer. You get a clinical picture, you do some tests, scans. Here you can see a child with a big lump in the tummy. A scan shows it's a lump in the kidney. You can't see there's little cores from a needle biopsy that go off to a histopathologist. They look at them down the microscope. These are pretty much small round blue cells. It's not terribly easy to know what they are, but on the basis of the pathologist's opinion, we will then decide what chemotherapy and whether or not a child needs radiotherapy in addition to an operation. But more and more now we're using biology in risk stratification. So the one that's been around the longest, apart, and I'm talking here mainly about solid tumours, Nick didn't go into the very extensive hundreds of different genetic changes known in childhood leukemia, but they've actually been around even longer than this gene that's called MYC-N. But this shows you your chance of surviving if you're a baby with neuroblastoma who's either got a number of copies of the MYC-N gene, that's the top line, so virtually everybody's cured. And if you have multiple copies, MYC-N amplification, you can see how bad the outcome is. And the test that you can do to detect this gene is called FISH, fluorescent in situ hybridization. You have a bit of DNA that matches the MYC-N gene, you label it with a fluorescent colour, and you can count the dots. So you can see at the top, a normal cell has 2 copies, and at the bottom you can see lots and lots of little pink dots. That tumour cell has got literally hundreds of copies of the MYC-N gene. But the problem with this sort of molecular analysis is it's very resource intensive. You have to have very skilled staff, you have to have very high quality tissue, usually frozen, and often the consumables. So these tests can cost hundreds, and sometimes at the beginning they used to cost thousands of pounds to do. It's not a reason, of course, making sure a child's on the right treatment also makes much more efficient use of health service money. Now, you've already seen this slide in Steve Lois's talk. So this is how molecular biology is coming into risk classification in medulloblastoma, childhood brain tumour. So already there are 2 subtypes described where we know what are the major biological signalling pathways. One's called Wnt and the other's called sonic hedgehog. These are pathways that are critical in normal development. And in fact, mutations in these pathways can be found in children with congenital abnormalities and not cancer. So cancer is part of a spectrum of how your normal embryonic tissue develops either normally or abnormally. Now, what the pathologists can do is they can turn this very complex molecular knowledge of medulloblastoma into quite a simple test. So if the Wnt pathway is switched on in your medulloblastoma, they can simply do a test called immunohistochemistry to look for a protein called beta-catenin, and here you can see the brown stain. And that indicates the good prognosis type of medulloblastoma. So a child with that test positive now will be selected in the current trials to have a reduced dose of radiation. And the basis here is that it will still cure the tumour, it's just as likely to cure the tumour, but it has less chance of causing problems with their education later in life. So modern diagnosis for a pathologist has now turned from the traditional triumvirate of clinical examination, imaging and some basic pathology to incorporating things like gene copy number. And at the bottom here you can see a whole genomic profile of a Wilms tumour showing gains and losses right across the human genome in this particular case. And we are just starting to learn how we can put all of these things together to give a patient much more personalised treatment. The other advantage of studying tumours in great depth, though, is that you can start to identify new types of tumours. Again, this is a bigger picture of it's showing copy number. So 0 is normal copy number. When it's above the line, you've got too many of a certain area of the chromosome, and when it's below the line, you've lost a bit. And this is, in fact, a subtype of brain tumour that's felt to be high risk. The thing is, you can have all these tools at your disposal, but what do they mean? So if 1% of childhood brain tumours have additional copy numbers of chromosome 11p, do we actually know what it means? And the only way that you start to know, should we actually be changing treatment based on this discovery, is if we work together, do the tests in the same way, treat the children in a standard way, collect all the data, and are then able to see what happens to all these different biological subgroups. And only then can you confidently say, right, from this point onwards, we should now recognise that this type of tumour is actually high risk, or maybe this type of tumour is actually much lower risk, and we've probably been given children too much treatment in the past. So biomarkers have enormous potential to help us improve how we approach risk stratification and how we pull out children who might benefit from new types of targeted treatment. However, there is a, if you like, fly in the ointment. It's this thing called tumour heterogeneity. So if you do a molecular test, you have to get a bit of tissue out of the patient. Usually the tissue's coming out anyway, because it's either a biopsy to work out what's going on, or it's an operation to take the tumour away. And there's always spare tissue that you can do this research and molecular diagnostics on. So here you see a Wilms tumour. We've been now investigating this very extensively in the import study that you heard earlier. So we're asking centres to take frozen tissue from multiple positions within the tumour. And here we've run a test that's looked at copy number gain on chromosome 1, the long arm, that's in the red circle. So you can see one specimen's got the gain and the other specimen hasn't. This is the same tumour, but it's growing maybe from subclones or maybe, so you've got heterogeneity. So if we'd based the clinical decision on the lower sample, we'd have said this is not a high-risk tumour, but we're starting to think, we're not using it yet, but we believe we've got emerging evidence that gain of 1q, as it's called, indicates a high-risk type of Wilms tumour. So obviously sampling is really important. The other thing is, how sensitive is your test? How hard do you look for the abnormality? Because it's perfectly possible, to go back to this one, that actually the sample in the lower line has got gain of 1q, but maybe only in 1 in 100 cells. And you wouldn't pick it up by this technique. But here's another gene that we know is bad news in many other childhood and adult cancers, a TP53 gene. Now this is found in diffuse anaplastic Wilms. You see we've managed to pull together 41 cases, which is a massive number because it's a very rare subtype. To start with, we just did ordinary DNA sequencing, and we found, to our surprise, only half the tumours have a P53 mutation. We then looked a bit harder with what's called deep sequencing. So you sequence that gene about 200-fold in each sample, and we found another 2 mutations if we looked with a much greater sensitivity. And then in addition, we had 3 tumours where the region that this gene sits in the chromosome, which is chromosome 17, is lost, but we couldn't actually find a mutation within the gene. But altogether, we can say probably about 2/3 of this subtype have got mutation in this gene. And what we found is that actually the children who have got wild type or normal P53, even though they've got diffuse anaplastic Wilms, are actually doing much better than we would have expected. And maybe we ought to be thinking about giving them slightly less treatment, whereas you can see the children who have got the mutation are still doing very badly, only about 40% of them are surviving. And really we need to think of a completely new treatment approach for this group. Now the other area where one can use biological imaging, biological markers is in imaging. So these are MRI scans, and on the left you can see MRI scans again of the tumour in the kidney. If you give chemotherapy, you can measure changes in that scan which might predict the histological composition of that tumour. So you might be able to tell you in advance of actually having the surgery what the risk group of that patient's tumour is, which means you could change treatment earlier and for the better. So this is what we're doing now. We have set up a very large study which everybody's participating in across the UK, and we're just adapting it to roll out right across Europe. The red arrows show we want to take blood and urine samples from patients on treatment for Wilms tumour during their chemotherapy. We want to look at circulating biomarkers. We want to do imaging research and, of course, study tumours in great detail, including doing a whole genome sequencing, which is something we're going to hear about later in this session. So finally, how does one deliver this sort of really complex study? You've got to give high-quality, standardised diagnostics using things like MRI scans, which not everybody does them in quite the same way. You've got to have a really good network of clinical collaborators who work with you, and this is how we work in the UK. Not all European countries are quite as well organised as this. But the other thing is you need the real expert knowledge of the disease areas. So I know an awful lot about renal tumours. It's quite a while since I knew as much as I would like to know about medulloblastoma, for example, because there's just so much knowledge in each disease area that to keep really up to date, you need to be thinking about it every day, linking in with your international colleagues who are in the same situation, running the clinical trials. So we are very organised in Europe. There are about 15 groups that work in specific disease areas, and we work very closely across Europe and with our colleagues around the world. So we are making sure that we're bringing the cutting-edge thinking to UK children, and as Becky may explain, sometimes it's a bit harder to get the cutting-edge drugs into the country in a timely fashion. So I think, just to conclude, that we want to, through biomarkers, both improve survival, but also quality of survival by giving the right treatment to the right patient, and that we really do need to work together to make sure that the trials are answering the questions that are most important to us all, and that the designs are acceptable and feasible.